Alright, so the last topic for biological molecules is going to be nucleic acids. Alright, so talking about DNA, RNA. Alright, so talk about proteins, we talk about the sequence of the amino acids in a protein. And these are determined by genes. And these genes are nothing more than segments of genetic code that encode for some kind of information. Alright, so in this case, you're talking about genes that are encoded by the DNA. DNA is a type of nucleic acid. Alright, now, just like the other biological molecules, alright, you have monomers for nucleic acids. Alright, these are referred to as nucleotides. Alright, so you can have basically nucleotides that are DNA nucleotides, and you can have RNA nucleotides. Alright, so there are two different types of major types of nucleic acids. Alright, so you have deoxyribonucleic acid, or DNA for short, and you have ribonucleic acid, or RNA. Alright, so what's the difference between DNA and RNA? They're both types of nucleic acids. What do they do? Alright, well, DNA allows for all this information. Alright, it's basically the instruction manual for a cell. Everything that cell does is some form or fashion encoded in the DNA throughout all its chromosomes. Right. Now, RNA. There are many types of RNA. Alright. You have RNA, like messenger RNA, that's basically a transcript or a rewritten copy of the information contained in the DNA. And then you also have other types of RNA that control and regulate gene expression. Control and regulate RNA expression. Alright, so when you think about the DNA, all the genes in the DNA are not always on. Alright, they're turned on and turned off. Alright, so they're expressed when they're needed. Alright, so gene expression. Alright, so basically talking about something called the central dogma of biology is this idea of DNA to RNA to protein. Alright, now the information contained in the DNA is going to be, in this case, DNA is going to be transcribed or rewritten into RNA. Alright, so it's like you copying, a friend's notes. Alright, and then the RNA, specifically the messenger RNA, is then going to be translated into protein. Alright, so this is like copying your friend's notes in English into, for instance, Spanish. Alright, because here you're going from the language of nucleic acids to the language of proteins, right? Which is a completely different language. All right, but you're going from DNA to RNA to protein. All right, so here, think about it. Your eukaryotic cells have a nucleus. All right, so what you're shown here is the cytoplasm of the cell. You're shown here you have a nuclear envelope or membrane, okay? And this nuclear membrane kind of encases your DNA, your chromosomes. And so the DNA and genetic information is kept separate from all other parts of the cell. Alright, so somehow you have to get information from inside the nucleus to outside. Alright, so how do you do that? Well, what you do is you take a gene that you want to produce as a protein and you copy it into messenger RNA. Alright, so you get a transcript. Alright, that messenger RNA then will leave the nucleus, alright, through basically a pore, a protein, a channel protein, into the cytoplasm. Alright, it interacts with ribosomes, which are basically protein factories. And here, your amino acids are assembled. The amino acids that make up that protein that are dictated by the individual sequence of nucleotides in the DNA. Those amino acids are arranged into your polypeptide, or your protein. Now, looking at the structure of your nucleic acids. Now, whether you're talking about DNA or talking about RNA, alright, you have the basic building block of your nucleic acids. You have your nucleotide. Alright, so your nucleotide consists of a phosphate, consists of a pentose sugar, and it consists of a nitrogenous base. Now, if you ever hear the word nucleoside, what that is referring to is basically the sugar and the base. All right, that's a nucleoside. Now, your phosphate and your sugar make up the backbone of DNA and RNA. Alright? And inside, kind of sticking out, are your nitrogenous bases. Alright, so kind of going back to this idea of proteins. Alright, recall that proteins are made up of amino acids, and all the amino acids have a similar structure. Alright, they're all the same except for their R group. Well, when you're talking about DNA nucleotides are all the same except for their respective bases. RNA nucleotides are all the same except for their respective bases. Alright, so that's a nice link between those two types of biological molecules. Alright, there's a standard basic makeup, right? A framework for these things. Now, notice how there is a polarity alright, to the molecule. Alright, so notice here, your phosphate group. Your phosphate group has a negative charge, alright, big negative charge, and it's attached to the 5th carbon of your pentose sugar. Alright, your base is attached to the 1st carbon. Now, your nucleotides. The nucleotides are attached from phosphate to your 3rd carbon on your sugar. Alright, so you get a nice little covalent bond that links these two together. Now, one end of the molecule, your 5' end, you have this negative phosphate group sticking out. It's a free 5' phosphate. At the very end of the molecule, you have a free hydroxyl group. Alright, this is your 3' end, your free 3' end. Alright, so you have a 5' end and a 3' end. Alright, so there is a polarity to the molecule. Alright, this is going to be important later on when we start talking about application or applications in terms of DNA and RNA technology, so biotechnology, and how we use the structure of DNA and RNA and proteins based on their chemical makeup. We can do stuff with it. We can manipulate it. We can visually see it. Now, looking at your nucleotides. Alright, so you have DNA nucleotides and you have RNA nucleotides. Well, DNA nucleotides, they have deoxyribose. Alright, they have, for their bases, they have cytosine, thymine, guanine, and adenine. Alright, so looking at deoxyribose, alright, look at the 2nd carbon atom in your pentose sugar. It has a hydrogen attached to it. Alright, this is in striking comparison to ribose, which has a OH group. Alright, so just one change in a functional group. You get a different sugar. Alright, now, looking at your nitrogenous bases. Alright, you have pyrimidines and you have purines. Now, your pyrimidines, alright, this is a bigger name, but a smaller molecule. Alright, so it only consists of one cyclic ring. Alright, so that's cytosine and thymine. Your larger nitrogenous bases, shorter name, purines. They're bicyclic. Alright, so guanine and adenine. Alright, so the way that I go about remembering this, alright, so think about purine, think about 9. Alright, well, if you look at adenine and you look at guanine, they both end with 9. Alright, so that should hopefully help you remember that adenine and guanine are purines. Okay? Now, what about RNA? Alright, RNA nucleotides contain ribose as their sugar. They contain cytosine, guanine, adenine, and uracil. Alright, now notice, RNA does not have thymine. Alright, but look at the structure of uracil and thymine. Alright, they're almost the same, except for your thymine molecule has a methyl group that's lacking in uracil. Alright, so again, the presence or absence of functional groups is affecting the overall structure and function of a component of a biological molecule.
DNA and RNA are nucleic acids. polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The 2 DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The 2 polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Alright, so talking about the assembly of nucleotides. Alright, so previously I mentioned that there is a covalent bond that joins one nucleotide to the next one. Alright, so this covalent bond is what we call a phosphodiester linkage. Alright, so here, looking at one nucleotide, you have a free 5' phosphate group here. Alright, so this is the first nucleotide in DNA, for instance. Or, sorry, this is RNA, because you get the OH group. And so, you know, this free 3' hydroxyl here, then OH. Alright, you have this phosphate group here with a OH. So guess what? This hydrogen plus this OH group, alright, you basically remove a molecule of water through a condensation, which is a dehydration. A dehydration reaction. Condensation, dehydration are the same thing. Okay? Now, now you have a covalent bond that joins this phosphate group, 5' phosphate, to the now 3' carbon in this ribose sugar. Alright, so this is a phosphodiester bond. Alright, it's a covalent bond. Alright, so the nucleotides are joined together through the component phosphate and sugar molecules. Alright, so that's why it's called the backbone of either DNA or RNA. Alright, so DNA. DNA exists as two polymers, polynucleotides, that are actually oriented in opposite directions of one another. Alright, so they're anti-parallel, and they actually form this helical structure. Alright, so again, this is showing you the polarity of, now granted this is a polynucleotide of RNA. Alright, but the same idea would apply to DNA. Alright, so you have a free 5' end here, free 5' phosphate. At the very end of the molecule, you have a free 3' hydroxyl. So there is a polarity to the molecule. Now, what's also important is that DNA and RNA are always synthesized 5' to 3'. Alright, so that always means that there is a nucleotide that's always added to a pre-existing 3' end. It's always in a 5' to 3' order. So it's always going to be in this direction, 5' to 3'. Alright, so DNA synthesis, RNA synthesis. It's always 5' to 3'. You're always adding a 5' phosphate to a pre-existing 3' hydroxyl. Now, there is complementary base pairing in terms of the DNA. Alright, so since you have two strands of DNA polynucleotides, and they're oriented in opposite directions, the bases of those polynucleotides are what interact and hold together the DNA as a double helix structure. Alright, so in this case, adenine is always going to pair with thymine. Guanine always pairs with cytosine. Alright, so you always pair a purine and a pyrimidine. Alright, so what's really cool is that later on we'll talk about the structure of DNA in a little bit more detail. But what we'll do is we'll also talk about the history of it. And one of the things that's important is the discovery of the overall structure of DNA, but also how the structure of the DNA is tied in with the process of how DNA is copied, how it's replicated. Alright, so RNA. RNA is single-stranded. Alright. You don't have thymine, you have uracil, and in place of deoxyribose, you have ribose sugar. Now, looking at complementary base pairing, it's always a purine and pyrimidine. Alright, so here you're looking at one strand of DNA here. And here you're looking at the other strand of DNA oriented in the opposite direction. Alright, so you got one strand here going top to bottom, one strand over here going bottom to top. Alright, so they are anti-parallel strands. Alright, so they're oriented 5' to 3'. Alright, DNA and RNA is always synthesized 5' to 3'. Now, guanine pairs up with cytosine, adenine pairs up with thymine. Now, why is it that these particular bases always pair up? Well, think about it. If you have two purines, alright, the larger structures, alright, there's not enough space or room for them to actually form chemical bonds. Alright, they're too close together. Alright, if they're two pyrimidines, alright, then they're too far apart to interact. Alright, so it's always a purine and pyrimidine. And what holds together the bases is actually hydrogen bonds. Alright, so between guanine and cytosine, you have a triple bond. Between adenine and thymine, you have a double bond. Alright, so these chemical groups coming off of the molecules, alright, the highly electronegative atoms, the oxygen and nitrogen, are interacting with hydrogen atoms in the other molecule that are also attached to a more highly electronegative atom. So you have hydrogen bonds that form between your respective bases. Alright, so DNA base pairs with another strand of DNA to form this helical structure. Alright, so you get the spiral. Now, RNA is single-stranded, but a molecule of RNA can actually fold in on itself and form a much more complex structure. Alright, so you have transfer RNA. And so you could have regions of complementary base pairing within an RNA molecule.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine.
Alright, so you should be able to talk about the differences in the structure of DNA and RNA. You should be able to talk about the differences in the function of DNA and RNA. Now, what if I was to give you a sequence of bases? Alright, so let's say you have adenine, thymine, guanine, cytosine, guanine, thymine. Alright, let's make this the 5' end of a DNA. Let's make that the 5' end. Now, what if I asked you for a complementary strand of DNA? Alright, well, here, this would be the 5' end. That'd be the 3' end. Alright, because remember, they're anti-parallel, but they're complementary. Alright, well, adenine pairs with thymine, right? Thymine with adenine. Guanine with cytosine. Cytosine with guanine. Guanine with cytosine. Thymine with adenine. Alright, so that's what the complementary strand of DNA would look like. Now, what if I asked you about the complementary strand of RNA? 5', 3'. Now, since RNA has uracil in place of thymine, wherever you would have usually placed the thymine, you place a uracil. Alright, so that's a U. Thymine is complementary to adenine, guanine, cytosine, cytosine, guanine, guanine, cytosine, and adenine. Alright, so that would be the DNA strand. That would be the messenger RNA strand.